This is the component 2 task book, and we are up to question 11, last thing on genetic engineering and applications. Here we have got a human cell, and here we have a bacterial cell. And here's our human gene that does something useful. It produces insulin. There's our human gene. Let's just follow it on this diagram. There's our human gene. There it is. So we followed that. Here's some bacterial DNA. There we are. Okay, there it is. We cut it, inserted our human DNA, and then somehow we put it back into a bacterium. So this bacterium has got its normal DNA and it's got this extra plasmid with our human DNA. And then this bacterium, then we grow this mitosis, it'll divide and divide, not mitosis, it's a bacterium, divide and divide and divide, and produce lots of insulin. So describe the process at A and B. Well, we can see we've cut this DNA there, and we've cut that DNA there, and we just need to describe and name any enzymes and anything else that we know about this process. Plus, we've already just done two questions in this task book that would help you with this. So the DNA is cut. So the DNA, or indeed the DNA is cut, and that is, from the human chromosome or the bacterial plasmid. That's the DNA that we've cut. And we could, what else do we know about this that we can put down here? We've got 4 marks, so it wants us to put some stuff down. What else do we know? We know the name of the enzyme, using restriction enzymes or restriction endonuclease, using restriction endonuclease. We've got 2 marks there for saying it's cut, using and naming the right enzyme. Always try and say the names of the enzymes. 4 marks, so we need to say some more information. Well, need to use the same enzyme, so the same restriction endonuclease, as this will give the same sticky ends, and so these will fit together, as this will give the same sticky ends as uses the same recognition site. So if we don't use the same enzyme, the sticky ends are going to be different, and so they won't form the hydrogen bonds between the exposed bases. So that's a typical question where there's quite a lot of marks there. There's almost half an essay of marks. And if you're just saying cutting and inserting, it's not good enough. You need to name the enzyme, name the other bits of detail about this. At point C, it's usual to insert a gene for antibiotic resistance. So actually, we stick in another. What colour should I do that? I have to do that in blue. So we stick in another gene that goes in there. Why is it necessary? Well, it's not actually necessary these days, but they tend to use fluorescent genes or something like that. But there is a problem of identifying bacteria that have taken up this gene, this human gene. Because this process here only kind of works kind of 1 in 100 times. This process here of sticking the gene in, that maybe only works 100, 1 in 100 times. And so the actual chance of getting a bacterium with the plasmid and with the plasmid that's taken up the human gene is 1 in 10,000. So you need to sort of isolate the bacteria that have actually got exactly what you want. So if you stick in a gene that you can use to identify whether it's all gone through nicely, then that's one way of doing it. We just need to sort of explain that now. So if we stick in the gene for antibiotic resistance, well, when we grow loads of these bacteria, only those ones will survive. So only the bacteria that have taken up the plasmid with the resistance gene and human insulin gene will survive. These will grow, the others will die. So we're left with just a pot of bacteria that contain exactly the gene that we want. Before that, we used to extract insulin from other animals. Extract insulin from pigs, which was very problematical. Least of which was animal welfare issues and ethical issues and religious issues. That's certainly a point, but also there were kind of biological issues, allergic reactions. It wasn't quite the same insulin. So lots of not a desirable way of doing this. And then also, as almost a concern these days, is lots of diabetes about, lots of insulin needed, and this is a great way of producing lots of insulin, very quickly. So we mentioned ethical concerns, e.g. animal welfare, religious concerns, and then we've got kind of the biological. The insulin was not identical previously, e.g. allergic reactions, and now can produce at a faster rate also. So you needed two, and there's kind of 3 areas you could have a crack at there. So a pretty straightforward genetic engineering question. Yours might be a bit more convoluted than that, but pretty straightforward for 8 marks there.